Welcome to the final section of Lecture 5 on synapses. We're going to talk about the fate of neurotransmitters after they've been released into the synapse and have potentially bound to receptors. It's important to get across the concept that neurotransmitters can't hang around forever. We don't want to send a signal and then have that signal always be on, always be on, always be on, because we could die. So we want that signal to just be a little burst. And if we want the signal to continue, we'll send some more neurotransmitters. So neurotransmitters can't hang around forever. So they just need to be a temporary thing so that we can control all of our nervous system functions. So there are 3 different ways that they can leave the synapse or kind of be gone from the synapse, called enzymatic degradation, reuptake, and diffusion. We're going to talk about the first 2 in more detail. Diffusion is a very minor contributor to how neurotransmitters get out of the synapse, and it just refers to the fact that they're just going to be they're just going to kind of move around. Some of them are just going to drift away out of the synapse entirely. So let's talk about the other 2 in more detail. Enzymatic degradation. So if we break that down, enzymatic means we're using an enzyme. Degradation means we're breaking something down. We're degrading it. We're breaking it apart. So in this diagram here, which is from the Hedges textbook, they've got, whoops, my animations were out of order. It throws me off every time. Okay, here we go. So we have a neurotransmitter, which in this case is acetylcholine. Here it is in its synaptic vesicles, exocytosis into the synaptic cleft. It just so happens that there's an enzyme. So anytime you see ASE on the end of a word, chances are pretty good that it is an enzyme if it's a word we're seeing in science. And so acetylcholinesterase is the enzyme that will break down the acetylcholine molecule. So they're showing you here that it breaks it down into its kind of component parts, acetate and, oh my gosh, acetate and choline. I don't care that you memorize that, right? What you need to know is that there's an enzyme in there that basically chews up the neurotransmitter molecule, so it's no longer an intact molecule, no longer capable of binding to the receptors and sending its signal. And so that is how the neurotransmitter gets taken out of the synapse or taken out of action, so to speak. Oftentimes, those little components, those little pieces of the neurotransmitter will get taken back into the cell, which will reassemble them and kind of recycle them so that now we have more neurotransmitter again for the next signal. Yeah. The other way is reuptake. So reuptake, in this example, we can see we've got dopamine is the neurotransmitter that has been released into the synapse, and you see it binding to a receptor here. But then notice there is a dopamine reuptake transporter. So this is a special pump in the membrane of the presynaptic cell that is going to take that dopamine out of the synapse and bring it back inside the cell where it can then be reused again. So it's kind of like take backsies, right? So it's like, okay, you can have it for a minute, now I'm going to take it back, and then I'll decide when you get to have it again. So that is reuptake. The neurotransmitter gets pumped back into the axon terminal so that it can be used again later. So there's a lot of clinical applications of these different ways that neurotransmitters get out of the synapse, get removed from the synapse, get inactivated. And so I'm just going to talk about a couple. One is things that block acetylcholinesterase. So let's remember acetylcholinesterase is the enzyme that breaks down acetylcholine. So if we block that, then we can't break down the neurotransmitter acetylcholine. And so acetylcholine, once it's released into the synapse, is going to stay there. It's going to stay active. It's going to keep sending the signal. The signal then cannot end. So this is how a lot of pesticides work. We do that to bugs on purpose. It kills them. It's also how nerve gases like sarin work, which are also highly fatal. Another example of this is there was a terrible accident in the country of India several years ago where the school lunch of some school kids was contaminated with pesticide. It was an organophosphate pesticide. And so what happened is all their synapses, when they released acetylcholine, they couldn't get it out. And 23 school children died died. It was a horrific accident. And so these things are no joke, right? So it is important to get neurotransmitters out of the synapse. They cannot stay there forever. An example of a clinical application for reuptake would be the medications that are called selective serotonin reuptake inhibitors, or SSRIs. So medicines like Prozac, Zoloft, Celexa, Lexapro, all of those medicines are in this class. And the way they work is that normally serotonin, which is what's being shown here it is in its vesicles, exocytosis out into the synapse, binds to its specific receptors. Normally, we have a reuptake, right? We have a pump that normally pumps it back up into the cell so that serotonin doesn't stay in the synapse very long. But what these medicines, these SSRIs, do is they basically block. So this blue bar is supposed to be kind of blocking that reuptake transporter. It blocks that from happening. And so then serotonin is staying in the synapse longer. And these medicines can be really effective for treating a variety of mental health conditions, including depression, anxiety, and others. They're not effective in everyone, but they can be extraordinarily helpful for a great number of people. Now, of course, I just told you that letting a neurotransmitter stay in the synapse longer can kill you, right? So you're like, wait a minute, what's happening here? Well, so obviously this is not preventing all reuptake from happening, right? It is just preventing some reuptake from happening. And if you overdose on an SSRI, if you take way too much, it can cause something called serotonin syndrome, which can very rarely be fatal. Serotonin is not, unlike acetylcholine, and it's not something that's likely to kill you if you have too much of it in your synapse, but it can be associated with a lot of symptoms of just excess serotonin, too much serotonin signaling in the synapse. So in summary, neurotransmitters have to go. We can't have them, right? The signal just has to be momentary. So we're either going to use enzymes to chew them up, or we're going to reuptake. We're going to pump them back into the axon terminal, or some of them just kind of diffuse out of the synapse. And so we talked about how acetylcholinesterase inhibitors can prevent the acetylcholinesterase from breaking down acetylcholine, which in the case of pesticides or nerve gases, those are very potent and it can be fatal. I will tell you that there are also acetylcholinesterase inhibitors that are used as medications, right? So obviously those are going to have a milder effect because we're not killing our patients. And then there are other substances, medications and other things that can block reuptake. So the example of SSRIs, there are also SNRIs, which is selective norepinephrine reuptake inhibitors. So we can use these medicines to kind of tweak and adjust the amount of neurotransmitter present in the synapse, and that seems to be beneficial in a variety of ways. Overall summary of synapses. Electrical synapses, we didn't talk about very much. We don't use a lot of them. That's where the neurons are directly connected by those connexons, like the adjoining rooms in the hotel. And so if one of them has an action potential, they all have an action potential, all the ones that are connected in this way. So it's really fast. It's all or nothing. It's near instantaneous. So that's pretty cool, but it doesn't allow for finesse. So most of the time we use chemical synapses. So the sequence of events here is that the action potential arrives at the axon terminal, opens voltage-gated calcium channels. Calcium comes into the axon terminal, which activates the snare complex of proteins. That snare complex of proteins pulls the synaptic vesicle, the vesicle filled with neurotransmitter, down to the membrane of the axon terminal, which then they fuse, and then the neurotransmitter diffuses out of the cell via diffusion, obviously, we call that exocytosis. So now the neurotransmitter is in the synapse where it will drift across and bind to its specific receptors that are specific for that neurotransmitter. If they happen to be ionotropic receptors, then they're going to open ion channels, and depending on which ions they're for in electrochemical gradients, we might get EPSPs or IPSPs. If they're metabotropic receptors, we could get opening of ion channels through that second messenger, or that second messenger could be affecting proteins or gene expression in the cells and could have a variety of effects on cell behavior. in the end, neurotransmitters gotta go. So for the most part, we're either going to use enzymes to chew it up and get rid of it, or we're going to reuptake, we're going to take it back into the axon terminal. And that's it for lecture 5 on synapses.